I was attending lectures at UCLA by faculty actively involved in treating cancer to try to expand my knowledge of cancer therapeutics. One lecture I attended was by a UCLA doc who described how common it is for cancer cells to develop in healthy people and how these cancer cells are usually detected and destroyed by the immune system. It is rare, but sometimes a cancer cell can learn how to evade immune surveillance. They go all Star Trek and use a cloaking device. Scotty, CTL incoming. Immune attack is imminent. Engage the Romulus cloaking device now. Hi, I'm Lenny Rome, the Vault Guy. I'm a cell biologist and a nanotechnologist. My passion is a common particle of the human cell called a vault. With the expert assistance of my daughter-in-law and unpaid executive producer Tara, I'm trying to explain what vaults are to non-scientists. Today, I want to cover 3 things. First, I'm going to tell you about cancer and a tactic that cancer cells use to hide from immune surveillance. Second, I'll introduce the lymphatic system and explain how lymph nodes work. Finally, I'll describe a new strategy to wake up the immune system so that it can identify a tumor. Stick around, and I'll tell you how we're going to use vaults to fight cancer. The lecture I attended at UCLA was given by a prominent immunologist and lung cancer specialist, Steve Dubinet. Steve works in an exciting area of cancer therapy called immune oncology. In his research lecture, he described this huge problem in treating solid tumors, which turns out to be the tricks that cancer cells use to evade our own immune system. Let's spend a minute on the origin of cancer. Cancer is caused by mutations in a cell's DNA that generally lead to uncontrolled growth of that cell. Cancer cells tend to have increased mutation rates, so as the cancer cell is trying to consume nutrients and grow, it's changing and often amplifying certain genes, seemingly at random. Some mutations turn on proteins that help the cells grow more, and some mutations amplify genes that help the cancer cell avoid the immune system. Even healthy people, as a normal part of their life, develop random mutations in their DNA that can turn a cell into a cancer cell. Most of these mutations are triggered by environmental factors like UV radiation from the sun. Fortunately for us, the human immune system is very good at finding these mutated cancer cells and destroying them. One bit of evidence that supports this hypothesis is that people who are taking drugs that inhibit or slow down the entire immune system develop more cancers than people who are not on these drugs. In Steve's lecture, he explained how a cancer cell can sometimes switch on genes that shield the cancer cell from immune system detection, allowing a tumor to develop. There are a number of different ways this can happen, but effectively, the cancer cell becomes unrecognizable to the immune system. One way to think about this is that the cancer cell can cover itself with proteins that trick the immune system in misidentifying the cancer cell as a normal cell. When I met Steve 20 years ago, he was working in what was then a new field called cancer immunotherapy, where different strategies were being developed to overcome the barriers set up by cancer cells to avoid recognition. Today, there are a number of these strategies approved by the FDA, and most are more effective than traditional chemotherapy, and they have fewer side effects. The 2 best examples are CAR T therapy and immune checkpoint inhibitors, like Keytruda and Opdivo. Steve had been working on a strategy to change the tumor environment to make it more welcoming to the immune system. What if we could make the tumor environment more like a lymph node? Steve asked. Lymph nodes are small glands throughout the body. They are part of the lymphatic system, which carries fluid called lymph between the body tissues and the bloodstream. The lymphatic system is an important part of the immune system. The lymph nodes filter lymph as it flows through them, trapping bacteria, viruses, and other foreign substances, which are confined in the node so they can be presented to the immune system. In the lymph node are signaling molecules that act as attractants and are called chemokines. The chemokine that Steve was most interested in is called CCL21. CCL21 diffuses out of the node, forming a chemical gradient. CCL21 attracts T cells and antigen-presenting cells called DCs into the node. Once in the node, DCs take up and digest the bacteria or viruses and use fragments of the invader to trigger an immune response. I gave you a brief introduction to this process in my last lesson. Steve reasoned that if he could make the tumor more like a lymph node, then the tumor, which has figured out how to block the immune system, might now attract immune cells into the tumor to overcome the block. CCL21 is an important chemokine as it has two functions. Not only does it attract immune cells into the node, but it can also stimulate immune cells that had detected foreign antigens. These are tumor-specific proteins that the immune system is looking for. To study CCL21, Steve's lab used a lung tumor cell line that can grow into a tumor if it is implanted under the skin of a mouse. They showed that multiple injections of CCL21 into the growing tumor stopped tumor growth and led to tumor destruction. Furthermore, they were able to show that the CCL21 treatment led to the production of an army of cells called cytotoxic T lymphocytes, or CTL. The CTL were attracted into the tumor, where they recognized foreign tumor proteins and attacked the tumor and destroyed it. One problem with treating a tumor with the CCL21 protein was that in order to be effective, the CCL21 needed to stick around so that it could sustain the signaling of the immune system. Since CCL21 is a small protein, it gets dissipated away from the tumor, or it gets degraded. Thus, multiple injections were needed, a very impractical treatment regimen for cancer. Steve needed a sustained release delivery system for CCL21, so he devised a plan. He isolated normal mouse DCs and taught them how to make CCL21. He did this by infecting them with a virus carrying the gene encoding the CCL21 protein. These mouse DCs could then be grown in Steve's lab, and he could show that they made CCL21 and they secreted it. When the CCL21 secreting DCs were injected into the growing mouse tumors, the cells released CCL21, and the CCL21 attracted immune cells to the tumors. The attracted immune cells reacted with tumor antigens and triggered a CTL response that destroyed the tumors. Just 1 or 2 injections of the engineered cells worked just as well as multiple injections of the CCL21 protein. These results encouraged Steve to try the therapy in his patients. This was a complex process that required the production of an engineered virus containing the gene for CCL21 that was approved for use in human cells. Furthermore, he had to develop methods to isolate DCs from his cancer patients, infect them with the virus, amplify their numbers, and inject them into their tumors. A small safety study was carried out in 2015 by Steve and his colleagues, led by Jay Lee, an oncologist at UCLA. The study was carried out on patients with lung cancer who had advanced disease that was not responsive to current therapies. The study showed encouraging results, indicating that it might be effective in humans. First, there were no serious adverse events. In other words, the treatment wasn't causing harm to any of his patients. In addition, over 50% of his patients' immune systems were activated, and cytotoxic T cells were induced. Overall, the initial trial was very encouraging, and it has led to a second trial that is still ongoing. However, there are still many problems with this approach that need to be overcome. The cost of this therapy is very high, estimated then to be over $150,000 a patient. Second, it takes weeks to prepare the cells, and finally, with this kind of cell-based therapy, it's very hard to control drug dosing, as engineered cells can be very inconsistent in their ability to secrete CCL21. So, what Steve needed was a way to avoid the cell engineering that has to be done for each patient. Perhaps a nanoparticle could be used to deliver CCL21. Ideally, one would want to use a natural occurring particle that would not trigger a negative immune response. I'm Lenny Rome, the Vault Guy. Join me in my next episode where I pick up the story and explain why vaults might just be the solution that Steve was looking for. As always, please remember to subscribe and give us a like. Just 1 million more subscribers and I can afford to pay Tara.